Okay, sir, you are now live.
Yeah, okay. So hope you can hear my voice and everything is fine.
Yes, sir. It's everything is fine, sir.
Okay, thank you very much.
Student is also joined, sir.
Okay. Sir, hopefully the audience will be near about 100.
No, sir, it's not 100.
Okay.
Sir, you should start your session, sir.
Okay. How many students joined?
Sir, it's 11 students are joined, and still it's
Okay.
more students are
In
available. Yes, more
in
students
between.
are joining.
Okay. Yeah, no problem. Okay. So shall I start?
Yes, sir.
Okay. So good morning to all of you. It's my pleasure to give a talk on fundamentals of research methodology across diverse scientific disciplines. Before I discuss on this topic, I would like to special thanks to the organizers from Bhakta Kavi Narsi Mehta University, Junagadh, for giving me this opportunity to discuss the research methodology and how to initiate the research work with the PhD scholar. So that's why I'm highly thankful to the organizers, Naval Kapurai sir also, and some other faculty members and university office bearers for giving me this opportunity. Let's begin with the important topics for the research scholar. So as you know, I will discover the many important points for these fundamentals of research methodology across diverse scientific disciplines. In my today's talk, I will discuss some of the introductions based on the research, then fundamentals of the research methodologies in terms of various natural products or maybe the organic synthesis, conventional method, and the variety of the method how for the preparation of different kind of novel scaffolds or the target molecule. The key important points and the question also I will discuss together, just like how to initiate the research work in the science, design and synthesis of the novel molecules with using different ionic liquid and dipyridtic mixture for the various heterocyclic scaffolds. And very important points I will also discuss with the scholars, that is green chemistry calculation. So the green calculation matrix is also important when we have to design a new molecules and we are claiming the sustainable approach. The little bit, I think, according to the discussions with the HOD of the chemistry department, some of the students may join from the life science. So little bit that the biological evaluations in terms of the anti-cancer activity, microbial and The other parameters also I will share with you by correlating the chemistry. Then microwave assisted synthesis for the certain molecules, click chemistry reactions, and the target isolations of the certain novel molecules from the natural species. Also, I will discuss the spiroheterocyclic scaffolds, monster analogs, which is already available into the in the market as a drug. So we'll show you that the some other novel monster analogs also, then some other heterocyclic scaffolds and the conclusion. So this is my today's outline talk for the research scholars. Friends, when we have to initiate the research in our laboratory, then what kind of parameter it must be required? That I will discuss with you. Can you see here how we initiate the research work? Let's see here with the full view how we have initiate the research work in the science. So, friends, there is nothing but the 7 key precursor steps which are very important to us when we have to explore the new science. Let's see here what are the requirements. So, we have to select the topic first. I will discuss with you the 7 important parameters for initiating the research. One, which is called the research interest. What type of research interest you are having and you want to explore the novel things. So research interest is very important to us. The second point I will discuss with you the literature review, how the literature review is very important for the students to get the novel idea, to find the gaps and to explore your topics in your own research. Then the third point, the problem statement is very important. At once, if you have to do the literature review, then you find a problem, you find a gap where the gap is there, and that gap things you identify, then you can explore the new things based on the finding gap from the earlier literature review. Then fourth point, that is the research objectives. The objectives is also very important to us. I will discuss with you one by one. Then the fifth one, that is called the research methodology, which methodology you are adapting and based on finding a gap from the literature reports. Then the sixth point I will discuss with you, that is the significance of the study, right? So that is also important. I will discuss with you one by one each point. And the seventh one is the feasibility, how you are able to do your proposed work, how you can satisfy, how you design a hypothesis and how you can satisfy it. So that feasibility also we have to check with our destinations, and based on that we can explore our research work. And of course the time frame, which is very important to us. Within the time we must complete our design works with the all parameters and data and other things. Let's see another important points. So as you know, first question arises with the 5 important words, right? So let us see here. There is a 5 important words which is shown here. There is a who, what, when, where, and why, and how. Let's see why. If I'm talking about the why, I'm choosing this research topic. Why did you choose this topic? What interests you have about this selected topic? And do you have any other opinion about the issue involved, right? As you know, many things the scientists reported. You did the literature based on the topics, okay? That is important. But so why you choose this topic? What is your interest? What is your target with your synthetic approach? Or any other things can happen to the life science also. What organ you are targeting? Which kind of study parameters you want to involve based on finding a gap on the specific issues. So that's why you want to select this topic, that is very important to us. Then another one is who are the sources of the information. I mean, you did the literature works, and then you see the resources that you received, sources of the information where you received, and you find it your topic, and you see the various survey on your selected topic, who might publish the information. So many of the people, they read the latest publications, and then they analyze these things, and they find a gap, and then they selected the problems, the lack of information is published earlier, and they have a new idea and they want to explore it. But the thing is, which journal you have selected as a reference, and which are the institutions they are involving with the research, and they are claiming the data, right? And they are connected with that the specific topics. Now, so that's why the information that you find it is also very important for the researchers. Now, another important question arises as a what are the most important questions about this topic, right? Is there a debate regarding the topic? Is there a wide range of subject and the viewpoint to consider, right? So that is also important for the researchers. Now, the fourth point I'm talking about, where does your topic matters most at the local, national, and international level? So, is the topic affecting the specific locations? When we talk about a natural product, then obviously it will be affected that the locations also. And the fifth one, when is was your topic important, right? So that is also a important question in case of if I'm talking about the natural product. Is this a current issue or on from the past? Do you wish to compare your subject to different eras? So, friends, these are the five important words which is very important to us for the selection of the topic, to collect the literature reports, and then we can find a gap, and then we'll choose that specific problem to fulfill the gap which was earlier there you identified from the literature. So that's why this the thinking and the kinds of question must we can include in our mind before selecting a topic. Let's see one by one. The one question is select a research area and topic. So for that, you have to select a specific research topic in a correct manner. And you can see that the importance of selected topic in a recent trends. And according to that, you have to select a topic. You have to select a research area or topic. Choose a subject that aligns with your own interests and with your lab expertise and the available resources to ensure the topic is a specific and researchable and then relevant to the existing body of the knowledge in your field, right? So many of the people they synthesize the kinds of new molecule in the laboratory but without any target, right? If we didn't have any other target, then why we synthesize this molecule? Why we waste our own money funding from the from the department, right? So we have to utilize our time with the specific chosen topic. That's very very important, and for that it requires the student interest and the guide interest for the selection of the such topics to explore the best thing with the chosen topic. So that's why the selection of a topic is very very important. Now the thing is how to choose your research topic. That's very important. You can see that the current trends and find out the gap, right, and the requirement, right? And based on that, you have to select a topic. So let us see here, whatever the research topic that you have selected, which is original and it is inventable and then intriguing. And then the recent and timely manner is also important and suit your specific interest and ignite your curiosity for the research, right? So at once you have to select the research topic based on the specific requirement, current needs to do the solution in the environment, to do the solution in the method development, to do the specific targets with the selected the organ from the living organism. So based on that, we have to select the research topic. Friends, another important thing is how to come up with a unique and good research topic. That is also a very important thing. So that we can keep in our mind for the selection of a topic. It must be reliable one. It must be research that we have to do it in a fascinating order in our laboratory. Based on that, we have to select a topic. Now the second point come over here, and that is nothing but to conduct a literature review based on selected topic. One by one, whatever the 7 points we have discussed in the one of the diagram, you can see this is the second point. So to conduct a literature review, that is very, very essential for our selected topics. So what we have to take care in that the literature review. So survey existing the scientific articles. You did the literature survey based on the scientific article which is available online or maybe with the library, either by referring the articles, either by referring the books and then the journal to understand the current state of the knowledge, right? So, and the requirement of the knowledge to explore the scientific idea and which is helpful to the society, which is helpful to the nation and worldwide. So we have to identify the research gap from this literature review. That is very important to us. And we have to avoid the duplication efforts. So we have to go with the careful literature review to find out a specific topics and the earlier reported works. You can compare it in terms of methodology. You can compare it in terms of the outcomes of the whatever the viability they claim, and what are the gap is there in the viability that you want to explore it. For example, someone have synthesized a drug molecule and they will try to explore this as a drug for the specific organ targeted. So then you can see that earlier drug which is available into the market, but they have several adverse effect. I mean the solubility issues, and then there is a several other side effect occurs, right? Rather than targeting a specific one organ. So that's why we will try to avoid a certain adverse effect. You can make your molecule water soluble. You can enhance the best efficacy of your drug molecule, right, as compared to the earlier reported drugs. So based on that literature review, we have to take care about those things, specific things from the literature as a gap, and then we'll choose this topic and we can satisfy them, and we'll move towards the hypothesis we can design for our selected topics. So third point I would like to discuss you friends here, that is called the define the research problem. After looking to this literature review, we have to define the research problem that is necessary to implement and to find a solution. And and based on that, we can formulate the hypothesis. So friends here, the clearly state the specific questions your research will answer, develop a testable hypothesis. You find a gap, and based on that you design a hypothesis, right, and a tentative assumption that you are predicting from the earlier report and can be tested through the experimentation and the observation. So that's why the finding of a research problem and to initiate the our hypothesis, and based on our predictions we can concretely design our project, and then we can initiate to explore that the your novel idea, which is the best as compared to the earlier reports. So let us see the another important points I would like to share with you. That is to prepare a research design and then methodology. That is very important. Looking to the literature review, you find a gap and you want to implement it. You want to design a hypothesis, but how you can satisfy your hypothesis based on your research design and methodology. So friends, here we must plan how you will conduct the study. How we can do this? We find a gap. We want to explore the new things in a synthetic approach, new things into the target as a certain drug molecule. So for that, it includes the type of a research, various type of research. We can explore it here in our laboratory. How we design that we have to think about based on the facility available in our laboratory. So in this methodology, we'll think about the experimental as well as the observations, as well as the data collection methods, survey, experiments, and the analytical techniques you want to explore it. Right. So based on that, we have to design a methodology, and you tried it to find a best methodology for the synthesis of any other organic molecules. And not only that, but the life science people, they can also understand whatever the drug molecule you design, and you develop a new molecules or the methodology by choosing a different methodology that it also explore as a novel things to synthesize a certain molecule. For example, you design a one synthesis that you want to carry it out at room temperature, and you can prepare a novel molecules, right, by exploring the chemistry. And at once you are preparing a novel molecules, your molecule must be soluble in the water, right? And then you can explore the in vivo and in vitro study in a nice manner, because there is a certain ethical guideline it must be required to find a target molecule. So that's why the solubility parameter, if our molecule having more then it is more feasible to explore that the in vitro as well as the in vivo studies also. Not only that, but there are certain other factors is also available with the research. At once we are preparing a new molecule, then we think about what is the molecular weight of the molecule, right? What is the log p value, isn't it? And what is the another parameters? So in that also you can explore your screening with using the computational approach also. So many of the people, they choose at the computational approach. For example, I want to work on this molecule. I want to work on this molecule. We have to select this molecule. We screen it on the computer with the in silico studies. Okay. We are screening with the in silico studies, and just by inserting a structures, you will get the data. So log p value is this, and some other parameters is this, right? So if you can have a different kind of the properties of your molecule that you received through the computational approach, and you match together with the certain bioabilities, there are certain other options are also there. When we have to do the in silico studies, it will give you the tentative idea your molecule will be target with this kind of specific cell lines, or it is also target with a specific protein kinds of protein. And based on that, the percentage also you can receive, and based on that you can target. Yes, my molecule may be target with this kind of the specific inhibitory targets. So life science people also take care about this, and according to that, you have to design your molecule with finding a gap, and you explore a new methodology. Definitely, your work will be fantastic, and you will target your molecules. So that is the one thing to design a research, to design a methodology is also important to prepare a new molecules. Not only in the case of organic synthesis, but in other there are so many other parameters are also there in which you can explore your own methodologies with your targeted, the bioabilities. Now, another points I would like to explore here, that is optical, the ethical clearance, if it is applicable. As I already mentioned you, our molecule is the best potent with the in vitro studies. We synthesize a novel molecule, we synthesize a library of the molecule, and we can check that the ethical clearance in terms of by doing this in vivo studies. That means, as I already mentioned, your molecule must be soluble in the water, or maybe soluble in DMSO with the 5%, 5% with some other solvents. They are eligible and they allowed with the ethical committee. That institute level or maybe the university level, there is ethical committee is also there. If life science people, they want to explore the in vivo study, we have to take permission based on selecting a solvent system. Your molecule will be soluble in that percentage of DMSO, which is allowed to do the experiment on the specific animal or maybe any other living organisms. So that's why this ethical clearance, which is very, very important to us. So we cannot harmful to the living organism. So that's why there is a strict rules which is available to do the in vivo study by taking care of our chosen molecule. The solubility is a must in water. Then it is a fantastic, and we can explore the very nice the applications or bioability. We can catch it if we have this one. So this is the fifth point in terms of the ethical clearance. To ensure your research follows the ethical guidelines, specifically, it involves the human participants or the animals. So this may require the approval from an ethical committee with the various university level or department level. Now the sixth point is a collect and the data analysis. Let's see here. So whatever the experiment we want to do it, okay. So during the screening of a molecule, during the practical approach, we can collect our data. For example, we have to synthesize one new molecules in the laboratory. And earlier we have seen that the similar kind of protocol, the many of the scientists, they explored it, right? So you can collect that earlier literature report also, and you can based on that you can find which are the gaps which is remain. For example, Lewis acid catalyst, can I use it? Can I use the base? Can I use the ionic liquid? Can I use the deputectic mixtures? So what kind of solvent system is prepared? What kind of the reaction condition it is prepared? Can be carried out this at room temperature? Can be carried out at the ambient temperature? Can be carried out above this 100 degrees centigrade temperature? Why this is required? So in that cases, we have to explore our own chemistry in our brain, and then we can utilize it for making this kind of novel molecule, which is very, very essential for us. So that's why the collection of a literature reports, collection of our own idea regarding selected topic, and then we can utilize it in our research work. I mean, to design our synthetic protocols, definitely will have a success in that cases. So collection of the analytical data is also important to us. So systematically gather the primary data according to your research design, and then one collected, analyze the data, and using appropriate statistical methods to interpret the findings. So that is also important at the last after doing the experiments and the so many screening in the viabilities. Then we can collect the data and we comprise the what are the best we did it in our laboratory and how we can explode it with the various journals for the publication or filing a patent based on what kind of finding you have. The another point is interpret how we interpret the finding as well as the report that results. So that is very important to us. Let's see here. We have to formulate the conclusions based on finding, based on experiment, based on the several kind of screening with your and whatever the data you have, that you can comprise it and you can analyze it, and you have to discuss with your guide and how your result agree or contrast with the previously work, right? So you can also think about your whatever the hypothesis you made it, and after experiment, whatever the data you received it, and you compare with the earlier, and you definitely find it if you search your molecule onto the Google with the IUPAC name. If you didn't find any other name of this kind of IUPAC name, then you will be surprised. And you must be happy because there is no report is found out with this molecule. Now let's check your molecules, how you design. You kept your protocol into the Google search, and you find if there is no methodology is available, then your work will be outstanding. So based on that, I'm talking about comparing with the earlier reports, designing your molecules and getting your data. You comprise it, and then based on that report, you can go where you want to go with the public. Either you are interested to go with the publication, either you are interested to go with the patent filing. So that is decided by the data, right? After collection of the data, you can decide where we have to go. If it is outstanding work, if it is a novel and you want to go with the patent filing, then you can also do it. If it is not, then you are interested to go with the good quality of journal rather than the ordinary journals, then you choose the specific Q1 quality, Q2 quality, or maybe the high impact journal. Not only I'm talking about the high impact journal, but I'm talking about you can check the quality of journals, and based on that you select the journals. But it is very important to you what it is, whatever the work you did it, whatever the data you received, and you can compare with the earlier literature work, then definitely you will get a confidence. My paper goes with this journal only, isn't it? So that kind of confidence you have it. But if your missing things is found out, right? I mean the lack of information you received from the literature, then also it may causes with your outcomes. So hope you can understand the seven criteria, which is very, very important to us to explore your own idea in a fascinating orders. Let's see the another points. Friends, hope you understand my explanation. Friends, suppose if we have synthesized our molecule in our laboratory, right? And we receive the final product after filtration. So after filtrations, there is a question of the purification, right? Question definitely arise. The purity, it must be required. The quality and quantity, it must be required for us. If you work with the pharmaceutical industry, QC department, they are taking care about the purity of your compound. It must be 99.9 percentage, right? That kind of outcome must be required with the standard pharmaceutical industries. And the crystallization, I will discuss a little bit based on the purification and the crystallization tools and the techniques, how we can use it in the laboratory. So isolation and purification of the organic compounds, like our molecule may be the solid or maybe the liquid, with a specific with a special emphasis on the chromatographic techniques. So purification, how we do the purification in our laboratory, what kind of facility we have in the laboratory. We may not have a facility to HPLC or maybe the automated flash chromatography somewhere else, but we must have a TLC, thin layer chromatographic system, okay, to catch the pure compound from the mixtures. Suppose if I'm talking about we have taken our final product after filtration, it is in solid form. Okay, sometimes what happened? The students, they didn't provide the enough time. student didn't provide that the exact molar ratio according to the requirement and the orientation and reactivity of the selected material for getting a final molecules, right? So that's why the equimolar, I mean, the you can see that the orientation and the properties of your starting material, right? So you can understand the property of the starting material. Based on that, you have to choose it. Okay, you can go with the reaction, right? How our molecule I'm preparing as a new by selecting this one, two, or three molecules at room temperature only. Why not? Why I'm using high temperature? Why I'm using the drastic condition? Why I'm choosing the very costly catalyst? Why not we prepare in a simplest manner? For that, it required to understand the important property of the three molecule you have chosen with having different functionality like electron releasing group and electron withdrawing group. And based on that, you have to design your reactions and you can catch your final product. And with the shortest time, you can prepare your 99% of the yield can happen. That would be fantastic, right? But what happens sometimes in the laboratory, also people they are doing, they kept the reactions and after completion of the reactions early, my okay product formation is there. They do with the filtration and then later on they will check the TLC. There are three or four compounds are there, right? What it indicates in the thin layer chromatography, if you, I mean, starting material, your starting material is three in your final product, you have a 4 point, four spot. You identify with the thin layer chromatography. What it indicates? It indicates that the three molecule is still there. One new point is there. It may be the intermediate. It may be the final product, right? But you can understand your starting material is not completely participated into the reactions and participate to convert into the 100% yield, right? So why not we provide a specific time? Why not we choose a specific molar ratio to get the 100% conversion rather than requiring any other workup procedures? And that's the great sign we must have with the good quality of scholars, right? So that kind of attitude you must take care about it whenever you have to initiate with your work. My point is to TLC, thin layer chromatography is very important to understand that how your molecule is pure, right? And what are the impurities are there that you can have it, and that impurity will be resolved if you have a facility like column chromatography. If you have a automated flash chromatography, you screen your that the four compounds is there in the TLC. You can screen with this the instrument and you can catch the simplest. I mean, isolated your pure compound, you can catch it with 99.9% of the yield, and that is the required in the pharmaceutical industry. Not only pharmaceutical industry, but if you want to explore your best outcome to target the organs at this time, it requires the major project purity must be required, because we will play with the in vitro cell line studies and then in vivo study. So purity is the major concern. So similarly, you can use the SPLC, so you can understand the percentage also. Then drying agent as well as the dehydrating agents, right? So your product is there, and you can see that sometimes it may not be in the dry form. So drying agents, somewhere you know very well in the gravimetric analysis, you did it what kind of drying agents and dehydrating agents we use. That the one parameters of the chromatography. So chromatography is the collective terms for the set of laboratory techniques for the separation of a mixture. You know very well. The mixture is dissolved in a fluid called the mobile phase. Your mixture, which is dissolved in a fluid, which is called the mobile phase, and which carries it through a structure holding another materials, which is called the stationary phase. With the TLC and other the chromatographic techniques, the various constituents of the mixture travel at different speeds and causing them to separate it. So how to develop a TLC is very important. Many of the people, they are not knowing. So friends, there is nothing but the 50% ethyl acetate and in hexane, right? You can see that the polarity and non-polarity, the combination, 50 plus 50, right? So you have a mixture, you dissolve it in particular solvent, and then you can see this is the solvent system, you choose it, all right? And then you check that there is a mixture spot is there, and you can see that there are three important points are there. I mean, the three product may be there, or maybe four product, right? So that is the thing, but how we develop a TLC, that is with the 50% the ethyl acetate in a hexane, we prepare the solution, and then we put this TLC onto the chamber, and then you can see that you allow them to run it and to separate with a very good RF value. Okay, so you know very well. Just I'm showing you. Another things you can see that 20% ethyl acetate and rest of the things with the and hexane, I mean the 80% and hexane. Then here you can see 25 ethyl acetate and rest of the thing is hexane. You will see here 25% of the ethyl acetate it requires, and then rest of the thing is hexane. Let us see here how the 1, 2, 3, and 4. Systematically, you find a RF value with the gap distance. So this kind of TLC, if you are able to develop in your laboratory, then you can separate it very nicely, isn't it? So please, it is very, very helpful to the all researchers, primarily in the in laboratory. So you must find a separation with your mixture, and you can identify the spot whether it is 1, 2, 3, or 4. That may be the four spot as a new product, or maybe sometimes it may be the unreacted portion based on the what kind of reactions and the conditions you carried out. Another thing, let us see here, calculating the RF value, how we calculate. I will not discuss that. I will skip it. You know very well, there is a solvent front, okay? And here we kept a spot of our mixture, the starting part here. And then you can see distance traveled by the solute, okay? Distance traveled by the solute and distance traveled by the B by the solvent. Hope you can understand RF value is equal to A by B. You will get the RF value. If we have a good RF value with a desirable distance, then we are able to separate this with the many of the advanced the instrument like automated plus chromatography easily, and we can have a pure compound. So another property of the commonly used plus solvents I'm talking about, the automated plus chromatography, I'll just show that the data so you can understand. The compound of interest should have a TLC, RF value 0.25 to 0.30 in the solvent system you choose it. And then the binary solvent system are two component with one solvent having a higher polarity than the others are usually best since they allow for easy adjustment of the average polarity of the eluent. And the ratio of the solvent determines the polarity of the solvent system and hence the rate of the elutions of the compounds to be separated. So the higher polarity of the solvent which increase the rate of elution for all the compounds, if your RF value 0.2, you will need a value of solvent 5x the volume of the dry silica gel in order to run your column. This is just for the flash chromatography I included here. So that is nothing but the column chromatography I'm talking about. This is another parameter is a column chromatography. And there may be the many type of column chromatography also there. We have a isomers also different. So then also we can have a different kind of the chiral column is also available. Let's see important words which is shown here that I will read it here. So column chromatography is an extremely valuable technique for the purification of the synthetic or maybe the natural products. So the compounds are separated by the column chromatography through the same mechanism as TLC through the different intermolecular forces between the component of the mixture with the mobile phase and between the component with the stationary phase. So you know very well, somebody you learn it in the analytical chemistry paper. A variety of the absorptions can be used as the stationary phase. And the silica gel, which is very polar, is the most commonly used in organic chemistry. So you can see that what kind of solvent system we have to choose it in the column chromatography also. Little bit I will show you in that manner. So as you know, friends, there is a polar component and a non-polar compound. So column chromatography is usually carried out with a mixture of the two solvent by choosing a polarity and non-polarity compound and their specific ratio. So commonly used solvent. So it is very important to you and you can keep in your mind. So the solvent, the common solvent that we use it mainly as a non-polar solvent like a hydrocarbon, as you can understand, and pentane and petroleum ether and then hexane. And in the case of ether and dichloromethane, very similar polarity, you can have it with this, okay? And then you can have this ethyl acetate. So you can understand the variations, whether it is polar, based on the polarity, we can have the purification of our compound by selecting a binary mixture. So the most common two component solvent system listed from this least polar to the most polar. Okay, you can see here ethyl acetate and hexane. You'll see that the polarity is enhanced. So when we talk about the ethyl acetate and hexane, which is most commonly used in the laboratories. So the good for the ordinary organic compounds and then the best for the difficult separation. Separation is a little bit difficult, but you will see that the standard solvent system that people are using in the TLC, then the methanol and the dichloromethane for the polar compound. We can use this solvent system for the polar compound. We must know the final product we have with this kind of functionality like OH and NH2 and other things. So if you have a polarity, then you can use this kind of combination of the solvent system to get the purity. Another one, the 10 percentage ammonia in methanol solution with the dichloromethane. So sometimes the most the stubborn amines of the baseline. So sometimes you can have a amine functionality, then it's very difficult to grow the specific the TLC and to separate out that the spots in the TLC and other things. So that's why we have a specific that solvent system. We use it for identifying those kind of functionality. For basic compound, nitrogen containing compound, it's sometimes useful or necessary to add a small amount of TEA, I mean triethylamine, as well as the pyridine. Okay, so I hope you can understand this. These are the different solvent system it must be required. So let us see here the height reached by the solvent front here. This is the solvent front, and you can see that this is our mixture. And you will see here red color and the blue color, and then I mean the light blue, sky blue color, and then this blue color. So you will see that how you spotted this point on the TLC, and there is nothing but the mixture of compound, how it is separated. I will not discuss that the column chromatography, and you can see in a simplest manner you have it to have a column tube, and you may use this particular solvent, I mean the silica gel, you make a bead and then with the mixture, and then you are adding a solvent. But here there is a huge wastage of the solvents that we have for the column chromatography, and it requires the huge time to separate these kind of the mixture, right? So that's why nowadays the automated flash chromatography, which is very much useful within the shorter period of time, and very a very less solvent system is required to separate those the mixture with the automated flash chromatography as compared to this column chromatography. So it requires huge time. It requires the wastage of these many solvents, and we can have this final separation. So hope you understand how we can separate this compound with the column chromatography to catch the pure compound, and you can separate these things. So I will try to skip this for the polar compound. So you can understand how our mixture, when at once you are preparing a bed with your mixture, and then you are adding a solvent system. So after half an hour, you will see that this much the separation happens. Okay, then 1 hour or maybe 2 hours, 3 hours, then 4 hours, 5 hours, 6 hours. You can see that how systematically we are separating A, then B like that. Okay, this is just for example to understand like this. Now let's see the isolation and the purification. It must be required. It is important part of the synthetic chemistry. As we know, friends, we know very well. We design our synthesis. We choose our starting material. That's why we know the orientation. We know that the mechanism, and we are expecting this much product. I mean, this kind of a molecule, we must have it after completion of my reaction. That is a known, and you have a particular idea regarding your formation of the final product. Okay, that is fine. But the thing is, let me try to explore very systematically. Friends, I'm requesting you choose a starting material in a systematic manner to carry out your reactions at room temperature. For that, it requires a certain functionality, and for making a CC bond, CN bond, CO bond, CS bond. So you choose a certain specific functionality, and you can understand how we are able to make a 2 CC bond, right, without using any temperature, isn't it? Without using drastic condition, without using a different kind of catalyst, right? So that is in our hand, and according to that, we have to select our starting material. That is very important and essential. I will call this protocol as a gap chemistry. What do you mean by gap chemistry? Gap chemistry is nothing but a group assisted purification chemistry. You choose your starting material, you choose to understand, you can understand the property of the starting material to get the final product. Okay, and sometimes it requires the solvent system. As you know, we have chosen three material for getting final product. So two compound is liquid and one compound is solid, right? What you have to do, how you can carry out the reaction. What do you think about the solubility of the solid compound, where it is soluble, isn't it? So based on that, you can choose the solvent system. If you understand your molecule is soluble with another two reactant, which is in liquid form, you just stir it and you check it. Your substance is soluble, then it's fantastic, right? So, so then you just stir it at room temperature and you check whether any other reactions is formed or not, right? So we have to design a certain reaction. We must carry it out at room temperature, and then we have to finish it. in a systematic manner in 100% of the conversion of yield. If it is not, then you can go nearest to this 90 or and then 80% of the yield must be there. But for that, it requires a strong support of the molecule to make the fantastic that C and CN bond formation and the cyclization to catch the final product rather than any separation. So if you are able a good chemist, you design a reaction, you after completion of reaction, you check the TLC. On the TLC, you must have only one spot, then you are fantastic chemist. Right? There is no need to separate the unreacted molecule. Directly you have a final product. That's the fantastic the chemistry, you have it. So you must have this kind of approach. Let me try to explain some of the workup procedures. Some of the people they design a reactions and they received a final product also. Okay, but after filtration they will find there is a sticky form is there in the funnel, right? Sticky form is form in the funnel. So what it indicates, so sometimes during the course of reactions there is eliminations of the water, there is eliminations of the CO2, there is eliminations of the ammonia, there is eliminations of the HCl, there is eliminations of the ethanol during the course of reaction. So I have seen that in the laboratory, some of the people they know very well this is my final product by eliminating this kind of solvent or maybe the reagent, maybe the reactant, or maybe the eliminated small fragments, right? Byproduct. So we must know that. Some of the people they forget about this. Okay, and then after completion of reactions, they after filtering they are having sticky form. They didn't have a dry. And in that cases, I also observe that with the apparatus when they kept it for the dry into the room atmosphere, then I have seen that there is a sticky mass, and I have seen that after 10 minutes there is a secretion of a liquid. Sometimes water is eliminated during the course of reaction. So some students they are kept they are kept the filter paper onto that and rubbed it, and then again they leave it, right? Then after half an hour again there is a water is there, right? What it indicates, there is incomplete reactions, right? They didn't provide it enough time, right? So that reaction cannot be happening, and you have this kind of observation. So unnecessary, you are wasting your time, you are wasting your product, and you are creating a problem during the course of reactions, right? So that then the another question arises: how we do the workup procedure? How we eliminate those starting material which is there with the product? I need a final product only in a pure form. How we can do this? Please be remember in that case, whatever the chosen reagent you have, where it is soluble. You check the solubility of your starting material, and according to that, you can give the treatment. But you must remember, your molecule must be soluble in different solvent as compared to the starting material. Okay, so based on that, you have to compare. My point is to friends, whenever you have to kept reactions in your laboratory, then you check. I mean, there is a one molecule which is soluble, two molecule is soluble, and one is a liquid. Okay, so during the course of reactions, after stirring, two molecule is not soluble in one reagent, or maybe they use the solvent system or catalyst. So there is a solid part is there. Okay, after refluxing, okay, you kept into the vessels, and for the uh, reactions at that time, you will see that after stirring, when you are slowly applying a temperature 30 degrees centigrade temperature 35 like that. So earlier you have seen that your two molecules are insoluble, but after 15 minutes or after half an hour, you will find one substance is soluble. Only one is remain. Based on the nature, you can observe. My point is to when you kept the reactions, every time you have to monitor. Your eye must be there with your flux. What kind of changes we have, and you can note it. Okay. Then after half an hour, you will see that your another molecule is also soluble. Now everything is a homogeneous solution. This is your another observation. Then you will find that after half an hour, again, you have a yellow color formation, isn't it? Then after 1 hour, then our 1 hour, 30 minutes, you will find little bit, the viscous solution occurs, and then the small particle which is spread around the flux. So your observation will tell you within half an hour there is a yellow color, within another half an hour there is viscous formation, another half an hour you have product formation. It started now. This is the key point where your product formation starts, right? So this is very important because sometimes you have a CC bond formation early, sometimes you have a CN bond formation early. So you can identify those, and in between you can test those things, what happened exactly. Okay. So that is nothing but the workup procedure is also important. I will not discuss much more about here because I have seen some of the observations in my laboratory that I shared with you. So product separation is also important, and the product purification, as I already mentioned that, and how much percentage of the yield is there, that is very, very important to eliminate the unreacted product from the mixture. Then the characterizations of the product and the further use and applications of the product is also important. Friends, the selection of appropriate isolations and the purification technique, we must use it. Now let me try to explain the chemical substances. So chemical substances, I mean synthetic or maybe the natural. If I'm talking about a natural, if I'm talking about a natural, then we are working with some natural species, right? So natural species with a living organism, I'm talking about with the one plant. Suppose if I have to select a one medicinal plant, you can have a root, okay, you can have a stem, you can have a fruits, you can have a flower, you can have a leaf, you can have a stem, right? So every part of the plant, they have a different constituent, they have a different phytochemicals, right? You can see that literature survey with one plant with their scientific name. You will get cluster of organic molecules. Not only we are preparing molecules into the laboratory, but God gifted us those kind of beautiful plant. They are taking care of the environment. And you will see that the number of organic molecule is there with the different plant species. So in that case, if I'm talking about, let me try to explain it a little bit beyond that. But if someone is interested to initiate the work on the natural product, you can select a medicinal plant. You can select that the specific, you go with the literature and you find a gap on which species of the plant there is a gap. Very, very few reports are there with the stems. Sometimes it is. Then you choose that the stems and you can make it natural drying, and then you make it in powder form at room temperature, and then you can go with the separations. Okay, I'll show you one or two example based on that. So natural product work is also important, but it requires the time and patience to explore the novel chemistry in that. Okay, so friends, there is nothing but whether it is synthetic or natural. There may be the two different kind of molecule. You have solid and liquid. Or if I'm talking about the natural plant, suppose sometimes you see some of the plant. Okay, you just see that the tulsi leaf of the tulsi, you rub it and you can take a beautiful odor. Okay, you can take you can there are so many examples you have. Okay, so in which case also you can see that some of the volatile organic compounds are there. As you know, in our home remedy, home kitchen, we have a spices, right? આપણે કહીએ ગુજરાતીમાં લવિંગ and તજ and other things. So if you can purchase directly the powder form of that kind of species from the market, then there is no specific order. It is in dry form and the volatile compound goes out. But if you have a freshly taken that species and you just rub it and you can add it into the food, or maybe the tea or whatever the things, and you can see that the beautiful taste because the volatile compounds are there. I will not discuss it. It requires a huge time to explore that the natural product. Okay. So friends, the liquid, you know very well the melting point behavior, sublimation, crystallization, amorphous, thermostability in terms of the solid compound. They may be decomposable and water soluble, or maybe the organic soluble like that. This is a little bit the property of the solid kind of molecules, and liquid molecules have a low boiling point and then the high boiling point, or maybe the volatile or non-volatile, or maybe the water soluble and the organic soluble, or maybe the gas form. So there are many techniques for the isolations if you want to go with this. So physical state of the samples, if you have a gas and liquid and solid and dissolved solvent and the dissolved gases as well as the solid. So amount of the sample micro and the macro as well as the nano, and estimated purity of the samples. I mean in terms of the simple mixtures and the complex compound. Isolation and separation method. Let me try to give you one example here. There are huge collections I have in terms of the slides. Let us see. Isolation and separation, how we can carry it out. There is nothing but the one of the Soxhlet extractions we have. If I'm talking about the, there is nothing but the solvent plus extracted substances here. Okay, so this is the instrument we use it for the Soxhlet extractions. So liquid and solid compounds and natural and synthetic products. Okay, it is very useful to carry out extractions to catch the different extract by choosing a different solvent system. using appropriate solvent with the limited solubility of the desired compound and no solubility for the impurities. And a small amount of the solvent is also required. And the product is separated from the solvent crystallization or the distilled recycling or the solvent is also possible. So, friends, there is nothing but a natural product. If you want to catch the certain species from the natural species through the extraction process, then this extraction method is very, very important to collect a water extract, to collect ethanol extract, to collect a methanol extract. Every extract, different fragment with a different solvent system, organic molecule have a different characteristic property to go with the different solvent system. And the fragmentation happens. As you know, one plant species, they have a 22 or more than 22 species are there, right? For example, ethanol, if you have to extract with the ethanol, you can separate the only 5 or 6 fragment or maybe the 4 fragment that goes with the extract of the ethanol. And you can have only four compounds as a mixtures into the extract. And then you can think about how we can isolate it and to catch a pure compound. That's the another important skill for the chemist. So let me try to explain one more example here. That is, we did some of the work. I will not actually discuss much more, but a little bit, a few slides I covered here to understand how we can work with the natural product. My point is to the many of the people they divert towards the natural products and to work on the natural product, which is very important. It requires the patience and it requires the huge time and the lots of effort is required to explore the specific findings, extraction and isolations from the different species. So this is our one of the outcomes. Let me try to explain with this graphical abstract. So friends, there is extractions of this. As you know, friends, this is the Lord Shiva's fruit. આપણે ગુજરાતીમાં બીલી કહીએ બીલી પત્ર ભગવાન શિવને આપણે ચડાવીએ, right? So there is extraction of the Lord Shiva's fruit. We did it when we cut it. Very important things, friends. You observe many of the people they use it. Okay, and you just cut the two pieces and then you just rub with your fingers and you take a nose, you just touch with your nose, you'll get beautiful odor. When you have this beautiful odor, what do you think about? There is one molecules. We proved it here. Okay. So friends, from this pulp, we prepare this kind of juice, fruit pulp in the ethyl acetate. We prepared it. We check the TLC. As you know, there are a number of states with the different kind of lights. You have this dark light, but we separated those molecules with a cluster of molecule by using ethyl acetate and then hexane with 50-50 percentage. Then you will see that from that extract, we have to grow a crystals. You can see that the beauty of crystals we reported here. That may be in crystal also we had a confusion. I will not discuss that the story, but that story will be there with this publication. So hexagonal shaped crystals we have, just like sugar. Okay, so square angle sugars or hexagonal or different kind of the shapes we have right in the carbohydrate chemistry also. So friends, there is nothing but beautiful crystal we grown it, and there is a hair-like crystal also. That is the same kind. We and then we identify this imperatorin as a molecule, and that single crystal we develop, and then we screen it with the XRD single crystal analysis. We prove this imperatorin. This is the first time we reported as a imperatorin from this bill fruits. As you know, this molecule, you take this molecule and take a order. You cut the piece and you can see that the liquid secretion and you touch with your nose and you can check the order. You will find that it's the same order with the crystal as well as the same order with the liquid that is found out. And that we proved it. There is nothing but the liquid secretion is nothing but like this. And then we, of course, explore that the various cell line studies also, anti-proliferative study we carried out with our one of the collaborators, in which you will find here whatever this compound we have as an imperatorin that can be screened with the in vivo study. Okay, in vivo studies and with the untreated cell, right? So there is only cell and with the DMSO, there is a standard. We didn't treat it with our molecule, but you can see here we treated our this molecule as a liquid secretion. We develop a crystal that will be treated with the cells. You will find here with the untreated cells, there are cancer cells are huge. But by treating with our molecule, you will find here there is a huge gap, right? So it is controlled over the growth of a particular cancer cell lines. So that kind of study we explored the biological people. Please be understand properly. Okay, you go with the article and you find that the certain studies. And if you have any other question, you please let me know. So friends, another parameters I'll show you here. There is a kinetic study of the HeLa cell lines, right? Then here also we treated with the HeLa cell line. So I will try to remove. I will not discuss that. Kindly see this crystals we grown it. Okay, So here you can see that there is a two type of crystals we are having. One is a cylindrical form and another one is a hexagonal form. So friends, there is observation is very important to us. How it is? Let us see here. So when we kept a single kept a solvent system with our molecule for growing a crystal, then you will find here there is a hair-like and there is a hexagonal shape. The crystals are there. We check the hexagonal case as a kind of cell of these crystals. Then we found out imperatorin is there. Then we thought that why there may be isomer as a hair-like crystals, and we checked it again. Then it found out it is also the same. Then we found out why it is happened, why the two morphologies are there. But for the development of a crystal, then we found out that somewhere, as you know, in a beaker, our solvent and the our compound was heated and evaporated the solvent and then kept it covered with the aluminum cover sheet, and then make a one or two small hole, and the slow evaporations happen. But one side, unfortunately, the cover is open up, and then you will see that whatever the hexagonal crystals are there, slightly you will observe this kind of they had like crystals by speedily that the solvent will be evaporated with the one corner and will have this kind of. So that's the another important finding we found out here. So I will not discuss much more. I hope you can understand the good quality of the crystals, and we separated earlier we received that they had like, but then we resolve it and again we develop like this kind of crystals. So crystallization is very important to us friends here. So I hope you can understand the cylindrical crystals we also observe the shape we have checked what type of it is. Uh, I will skip this crystallization so you can understand here. So image of the optical microscope, this kind of crystal hair-like crystals, what kind of shape and size we have that we also observe. So there are number of crystals we prepare. So crystallization is also important to us. That is, my one of the student Ruturaj Singh Wala from the Junagadh. So he developed this cluster of molecules, crystal, and here Divyang Patel and here also Mayank Sharma. Earlier they prepared and the beautiful shape of the crystals they prepared it. So friends, there may be the many methodology for developing a crystal. You can't understand with the evaporating dish. This is the whole evaporating dish is full filled with these crystals. That was the beauty. One day I earlier we observed to grow the crystals from a substance which is soluble in DMSO only. Use the slow solvent diffusion methods such as a layering a non-DMSO solvent or an anti-solvent like diethyl, diethyl ether into the bottom of the long container and then slowly pipetting the DMSO solution on the top. So there are another methods which is shown here. Another method is it allows the layer to slowly mix or use a vapor diffusion techniques. But this is another method as a vapor diffusion techniques by placing the DMSO solution in a closed environment with a more volatile miscible solvent like diethyl ether. So another option is the slow evaporation that we did it here. Measure of the our developed crystals with the slow evaporation techniques of the concentration of the DMSO solution at the room temperature, which is a different solvent system. So friends, I think I will not discuss much more about that the crystallization techniques we have that I'm skipping here. So friends, here another points I would like to mention you here. So hope you learned that the many books, okay, in the heterocyclic chemistry. In the heterocyclic chemistry, you learn the many books in which you will find that the role of a catalyst, okay, role of a catalyst, as you know, specific catalyst. But I'm talking about today here is ionic liquid and the volatile organic solvent. Why volatile organic solvent is a replaceable and it requires to replace it anyhow with the ionic liquid because of the causing of these volatile organic solvents. As you know, it increases the pollution as well as we cannot be easily separated from the desired reaction product and the difficult to recycle it, right? In view of the awareness of the weakening environment, researchers are directed on developing an alternative environmental friendly solvent system to replace the traditional volatile organic solvents with the contest interest of ionic liquids as a green solvent and resides in their extremely low vapor pressure and the high thermal stability. And that's the characteristic property we have to take as an advantage, such as the easy of control and product recovery and recyclability. That's why many of the people they explore the synthetic protocol by using different ionic liquid as a catalyst, but certain limitations. Ionic liquid will be replaced by the volatile organic solvents. So friends, you can see that the characteristic properties of the organic solvent as well as the ionic liquids, in which you will find that the ionic liquid is best as compared to organic solvents. Okay, so I will not discuss much more, and I will not discuss that the other ionic liquid and other parts. But you can, I will explore here what type of ionic liquid we have. Ionic liquid have specific features, as you know. Just I will show you that the ionic liquid is the generic terms for a class of a material consisting entirely of the ions and being a liquid below 100 degrees centigrade temperature. If they are a liquid at room temperature, we call them as a room temperature ionic liquid. So friends, that is the thing, but N is here. And you compare with the ionic liquid, there is a symmetrical anion. There is a symmetrical cation is there in this ionic salt. But ionic liquid, you can see here, there is a symmetrical ion is there, and then asymmetric cation is there. Friends, there is a huge heterocyclic chemistry is there. Huge heterocyclic molecule is there. We can take as a, with a nitrogen containing compound and we make a salt. Okay. So with a different kind of the symmetrical anions, and we'll prepare a different kind of ionic liquids. I will skip this, the different kind of example, and but I will show you this. The advantage of the ionic liquid, mainly it requires huge time to explore this, but just the advantage of the ionic liquid is to easy separation, very low vapor pressure, no inflammable, no flammable, non-flammable substance, high thermal stability, and then high mechanical stability, electrochemical stability, low toxicity, and non-volatility. And you can see that there is a broad applications which is explored: biological, analytical, and the solvent catalysis, physical chemistry, electrochemistry, and the engineering. And even though with the natural product separations, also people they are exploring, they are using the novel ionic liquid and go with the separation of the natural products and to catch the specific and the desired product, or the selective molecule can be picked up by using the ionic liquid for the separation of the natural species. Well, in terms of reactions, I will show you a little bit. Sorry. As you know, friends, the water is nothing but very good lubricating agents, okay, for our body, for our living organism. Okay, friends, I'm talking about the multi-component reactions in organic chemistry, how it is a powerful tool for the synthesis of many diversified organic molecules. Let's see here. As you can see that the importance of the multi-component reactions, high atomic economy is there, easy conversions are there, then the easy of executions, as well as the shorter reaction time is there, and reduction in a number of steps. Friends, you know very well, suppose alkaloids, suppose terpenoids, suppose you can have vitamins. If you want to synthesize in laboratory, you will see that there are many total syntheses and semi-total syntheses are there. It requires number of steps, it requires number of reagents, it requires number of catalysts, several consumption of the time and the chemicals, right? Like that. So here we will try to reduce that the steps. Okay. So as you know, if I'm talking about in the alkaloids also, you can see that Robinson and that scientists, they explored that the one of the important natural product is alkaloid in a one pot rather than the several 7 to 8 step. One of the scientists who explored, we synthesize this molecule. But you will see that Robinson and that team, they synthesize with the one pot three component reaction, and directly they receive that the close to that the final product, right? So that's why multi-component reactions is very important to us to reduce the steps and then to reduce the time also to get the desired product in a systematic manner. If you can take if you can maintain the gap chemistry for selection of the starting material, and it also allow us to assess the complex of the diversified molecules. We can prepare it by using this. Now, how we can do this? What kind of protocols we use for the multi-component reactions? Friends, there are huge scope we are having. Just for I'm talking about the acid and base catalyst we use for the multi-component reactions. Multi-component reactions, generally you will see that for the cyclization with having two CC bond, one CC bond, one CN bond, two CN bond, one CC bond, CO bond, CS bond. For making a cyclization, many of the people they use different kind of catalysis for the multi-component reaction, like acid and base catalyst. We may use the ionic liquid, as you know the importance as compared to the ionic liquids. Then we also use the organocatalyst. We may use a gap chemistry, group assisted purification chemistry. We can apply for this multi-component reactions. We also use a microwave irradiations for our selected molecule in a reaction to get a desired product. Then nanoparticle, as well as the ultrasound techniques and additive free, and then deep eutectic mixture, is also there. We prepare a novel lipotectic mixture to explore it in a cyclization and to catch a final product. That's a wonderful chemistry, and we can maintain the certain the green chemistry principle also. So just I will give you some hint. As you know, they are the PhD scholar. They initially registered in the MK in this university, BKNMU University. So friends, there is nothing but the 12 principles. Somewhere you learn it in the books or maybe in the syllabus somewhere. So 12 principle. If we are not able to satisfy whole the 12 principle, but somewhere we satisfy the 8 protocol for making a for designing a new molecule by applying a novel approach, then it would be fine. So, like a waste prevention, atom economy, and less hazardous chemical process, designing a safer chemicals and a safer solvent system, auxiliaries, and then energy efficiency and renewable feedstock and the reduced derivatives, catalysis, and designing for the degradation, real-time analysis for the pollution prevention, and safer chemistry for the accidental prevention. So we will try to take care about this 12 principle and will based on that we'll design our reactions, right? So friends, again, one more parameters I would like to share with you. I think we have 15 minutes more. So solvent selections, these are the solvent which is preferred in a green chemistry. Water, acetone, then ethanol, 2-propanol, 1-propanol, ethyl acetate, isopropyl acetate, methanol, and 1-butanol and tertiary butanol. Whereas another solvent system is listed in this another column that is usable, right? If there is no option and it requires you go with this solvent system, but you can't with this undesirable solvent which is shown with the red color, right? So that is harmful for the environment. That's why it is avoided to use it in the reactions, in any new reaction. So hope you understand this. Now, I will not discuss much more about the ionic liquid, how it is useful to, extract and isolate that the specific, the organic molecule from the alkaloid series, terpenoid, flavonoids, natural mixture, aromatic compound, others, right? Phenolic acid, ligand, and lipids, right? So you can see here, this is the diagram which is shown here. This graphical presentation, which is clearly indicated here, how the ionic liquid used as an extractive solvent as a function of a different ion combination, right? So ionic liquid, there's a plus charge and negative charge. Negative charge with the anions, and then plus charge with the specific hetero atom containing that species like benzimidazole and other thing, pyridinium salt, like that. So hope you can understand the different kind of combination leads to uh, separate that the molecules from the different plant species, right? So this is just for your example how the natural compound can be extracted, right, with using ionic liquid. So the majority of this the ionic liquid is used to extract the alkaloids. There is a 24 percentage. Then terpenoid also there is a 14 percentage, like that. So hope you understand the importance of the ionic liquid in the natural products also. So I will skip this, which is very important, but I will not discuss. Now, friends, I would like to. We have at 15 minutes, so within the 15 minutes, I will try to explain you how we design a new molecules. So, friends, let me try to explain here. So, this is aminopyrazole. This is our aldehyde, and this is our 1,3-dione. You know that the active species here, right? Active methylene. You can understand the carbonyl carbon atom, the electrophilic characters. There is a amine. Okay, let me try to explain here. So, friends, here there is nothing but ionic liquid is used, which is called the 2-hydroxyethyl ammonium acetate that we use as the best ionic liquid for catalyzing this these reactions and to have this product. So, friends, there is nothing but we'll have a 2 cc bond formation. Here, 1 cc bond formation. Okay, we'll have this kind of cyclization to have a pyrazole pyrimidine, right? So, that kind of core, or we may call it as a quinolinone core. Okay, so, friends, here we prepare this number of molecule. So you can understand this is a different octet diagram of our crystal. We draw a 4 crystals and we reported this, and this is the octet diagram. So I'm just talking about the important chemistry. What do you think about the orientation and reactivity of the aldehyde? This the amine with having this pyrazole core and then 1,3-dione. So I will show you this one. So friends, we prepare this one, and we use a different kind of ionic liquid. Best I will not discuss much more about, but you can optimize that reactions by selecting 3 components with certain condition. As you know, there is a 2-hydroxyethyl ammonium acetate. Somewhere it is you can see that the lactate form. There is a another form is there. Okay, So these are the different kind of ionic liquid we use. But we found out the best ionic liquid is 2-hydroxyethyl ammonium acetate as ionic liquid, as they have a dual characteristic property. Either they will try to abstract the proton from the carbon, or they may donate the proton to the specific things and will have this reaction. So 90 degree temperature is required. 2 hours, within the 2 hours, we'll have majority of the product. This is the percentage yield which is quoted by taking this kind of different condition by choosing different ionic liquid. So friends here, okay, this is very important for you. I'm just now very few time is there, so that's why I will give you some orientation. Okay, let us see. So friends, as you know, according to the literature, we designed this molecule to prepare this kind of 2 CC bond and 1 CN bond. But friends, here looking to the literature reports, we found out that you can see here this molecule NH2. NH2 attached with the carbon of the carboxylaldehyde, and there is a active methylene. We have this kind of formation. In the report, we found NH2 will be interacted with the carbonyl carbon of the aldehyde to make a CN bond. But in our case, you can see that where NH2 attached here, there is nothing but the possibility this NH2 or this carbon atom. Let's see here, NH2 is attached on the carbonyl carbon, whereas NH2 attached with this carbonyl carbon of this 1,3 ion. Okay, whereas here NH2 attached with the carbonyl group of the carboxylaldehyde. So selectivity, how it goes. So according to the literature report, they are claiming this kind of cyclizations happen. But in our case, we receive this one. So that's why, friends, you go with the orientation and reactivity with your chosen molecule, and you draw the reactions and final product according to your expectation and knowledge. But you also think about diverse way. Can it happen here? This is the carbon atom where we have a attached with this carbonyl carbon, right? You think about the another probability also. When you think about a little bit different, then you will get a novel chemistry. Okay, but you can correlate them as a orientation and then reactivity, and then you can predict your molecules. So hope you understand here. There is nothing but this is one parameters. I will not discuss. I will give you some another examples also. So we didn't receive this, and this is the final product. So we prepare this different ionic liquid. At once we use ionic liquid. If we use a catalyst, then also you can check with this recyclability. Green chemistry principle, recyclability is a very, very important tools. Okay, so one time you completed the reaction and get the final product, but you have to collect that the ionic liquid and then again you screen it. Okay, reuse it. So you can see here the reuse of this ionic liquid up to 77% of the yield we received up to 4 run. And you can also see that the time is expanded and the percentage yield will be decreased. So I will not discuss that the mechanism also. So you can go with our publications and the supplementary file, you will get the mechanism. Friends, you can see that how the beautiful crystals we grow in our laboratory with a slow evaporation method, golden color, right? So beautiful different structures, as well as the orbit tape diagram and the packing rearrangement we have. So then, whatever the synthesized molecule, will send it for the different activities. So you will see here for the uh, tuberculosis activity with the H37RV. So you will find here this is the standard drugs, and our one molecule which is best potent with the D15 molecule with having this specific microgram per MIC value, I mean microgram per ml, as compared to this rifampin as a standard drugs. So similarly, antimicrobial activity is also there with a different our structures, and comparing the standards, we have only one compound is the best potent, that is a 12.5 as compared to ciprofloxacin as well as the nystatin, which is available into the market. So in anti-proliferating also, these are some data. So this is the conclusion for that. So hope you can understand this protocol. I will give you some another example also. Friends, here, this is our another publications in which you will find the pyridine acetate, pyridine 2 carboxylic acid that we use as a catalyst for making this 2 CC bond and 1 CN bond. As compared to earlier one, you will see that how we have this. Okay, but another protocol that we use, we also use the ethanol also at 60 degrees centigrade temperature within short period of time. We received, then you will find the publication here. Let me try to explain another publications here. So friends, here you can see there is nothing but we have this molecule and substituted 2-cyanoacetamide. And here you can see there is nothing but the piperidine acetate that we use with this malonitrile and aldehyde. We prepare this and then we use it. We'll have this kind of product. But you can see One more thing we found out here, there is a pyridine derivatives we prepared. Okay, but here in this case, suppose if we have to take a phenyl ring system here and the naphthyl, then it is possible. But if we have a anthryl group system with the aldehyde, then due to that crowding, we didn't receive this product. Okay, so hope you can understand here. So this kind of simplest the chemistry we did it in our laboratory, and we moved with the publications. I think we have 5 to 7 minutes more. Let's see here one more example and the protocol I would like to share with you here. There are huge examples we have. So deep eutectic solvent. Okay, so deep eutectic solvent is little bit advanced as compared to the ionic liquid for this different kind of the product formation. So friends, I'm talking about here. According to the literature, we found out this kind of 2CC and 1CN1 formation by using trimethyl glycine betaine base and oxalic acid. This is the deep eutectic mixture we prepared in our laboratory by taking natural source as a trimethyl glycine betaine. And with the 20 percentage of the mole, we are thinking about we must have this product. But friends, when we check our product and with the mass, we found out a huge mass because there is a more mass is used. We found out the mass value is higher as compared to this one. So as you can see, the 2 moles of the aldehyde is used to make a spiro molecule, and it's a novel thing we found out, and we published with the ACS Sustainable Chemistry and Engineering. There were 14 molecules we prepared it, and we used a TMGB-based sustainable catalyst, and we didn't use any column chromatography, and we have this the low E factor and the environmental factor according to green chemistry principle. We have below the one value, it indicates that the better reaction protocol and that is compatible with, that is mashed together with the sustainable approach. Regioselective synthesis we carried out, and 99.7 percentage of the purity we monitored with our synthesized molecule as a spiro molecule. Let me try to explain rather than this one. Okay, so friends, there is nothing but the one per one decomponent reactions. We use this root beat, you know very well. There is nothing but the betaine contain is there, right? In present scenario, this multi-component reactions with this one, it's very advantage. In 2019, we published this work. So friends, there is nothing but the importance of the TMGB, I mean trimethyl glycine betaine based catalyst. So TMGB base is a natural byproduct of the beet sugar, as you can see. Now the amino acids, there is nothing but the amino acid derivative is applicable in a food supplement also for the animals as well as the personal care ingredient. You will find that role. Now, the treatment of the non-alcoholic steatohepatitis as well as the enzyme stabilizer, it is also used. We utilize this, we prepared it in a laboratory as an ionic liquid, right? So, hope you can understand this one. And there are several comparisons we did it, and with the earlier report, this is the earlier report how the people explore that the kinds of product formation, and we have different as compared to the earlier report. These are the reference earlier we shown, and we compare with our work. This is our best work, and that is completely novel as compared to the earlier report. And we have a diversified product as a dispiro, as a spiro molecules. So that is the things. These are the optimization protocol. I will not discuss. I think we have 5 minutes more, and I would like to discuss some other things rather than the mechanism. We developed the crystal. And one more thing I'd like to share with you, as you know, the green chemistry principle, which is very, very important and essential for us. Okay, so friends, we completed our reactions. We know that the starting material, how much mole and molecular weight I mean, the mass we have taken and final molecule percentage you have. So we have to calculate the E factor according to the green chemistry principle. E factor can be calculated using mass of the raw material and then the mass of the product. Upon mass of the product, you will get the E factor. If you receive the E factor below the one, then it would be best and best fitted to the green chemistry. And atom economy and reaction manner efficacy also carried out in this way. You will find that these are 10 things, and there is nothing but the green matrix is calculated, and you will find here this green line, it indicates that near about 0.13 to 0.5 is the best E factor we received for these desired reactions. So these are the some in the anti-proliferative activities. Okay, so there are hello organizers. Hello, hello. How much time we have? Hello,
Yes, sir.
hello, friend. How
Yes,
much
sir.
time we
Yes.
have? Let me know the
Sir,
time.
it's
So
5 minutes. 5 minutes for
yeah,
five.
okay. So I will try to finish my lecture within 5 minutes. So we
Sure.
also carried out the number of reactions based on the glioblastoma cell line. I will not try to discuss with you. Just I will show you these reactions here. So friends, there is nothing but here also we take aldehyde and there is a lawson is the molecule. Okay, and then we treated with this molecule to carry out the final product as a linear and non-linear molecules that we prepared with the new and novel work that we reported here in the ChemCog. So friends, there is nothing but there are so many literature review. You find a literature review in which you will find that the importance of the quinoline, how it is, and how they explore that the kinds of activities. Even the drug molecule also you can have the quinoline nucleus as a component, okay, and how we check the literature and we design that molecule with having the quinoline core. You will see that friends, these are the different reaction protocol people applied, and then they want to make a cyclization, but they didn't receive it, and they have this kind of uncyclized material. But friends, you can see here, according to the earlier report, we assumed that this kind of product must be there, but we didn't. And we have a new different isomer. I mean, the linear and non-linear molecule, we received it and we separated it. I will not discuss that. Okay. So these are different scaffold. We prepared it in our laboratory with the linear and non-linear molecules. These are the mechanism, LCMS, how we separated, how we correlates with this, and our structure is there, like with the HMBC and HSQC. I will not discuss. There are several cell line studies and other reports. Due to lack of time, I will try to skip it. But friends, here I'm talking about. So our two molecule out of this 17 or 20 molecule, we have only two molecule that is the best potent with the 2.9 micromolar and 3.3 micromolar as compared to the drug available into the market that is called temozolomide that is 50 micromolar. Our molecule have this one. So you can understand the importance of our synthesized molecule with having this base finding. So here also you can see, I think the time will be over. I will try to skip it, but you can see here this is the Viginelli product. Viginelli reactions, you know very well how we prepare this. We prepare this molecule, and then by interacting with this in presence of methanol, room temperature, and then we'll have this kind of diversified stereospecific synthesis we also carried out. I will try to skip it. But friend, one more thing I'd like to tell you here. There is nothing but these are the molecules, heterocyclic core, right? Many of the people they synthesize. This is the literature report, friends, where they use this chloroacetic acid and the same kind of this kind of material. Here also they use a different kind of ester. Let's see our approach. Instead of acid and ester, we have this molecule as a new starting material that we have chosen to make this kind of cyclizations and diversified and novel product. So hope you understand this. And we also carried out a gram scale synthesis at the laboratories with using this protocol. I will try to skip these slides. There are huge things to explore it. It requires 1.5 hours more to discuss very nicely and in a fascinating order. But taking much more time. Sometimes if I have a time, then definitely I will discuss. There are huge reports we have. So I will skip this and move towards the another one. So I will try to skip this because the time is the limit. And I will try to close it here. Just I'll show you the last slide. Okay, there are huge things we have. Sometimes I get the opportunity, then definitely. Okay, so this is friends. These are the reference. Okay, we have with our works. So friends, recently we published our paper in the green chemistry. It's very important things, as you know. The click chemistry approach earlier reported by the scientists in 2023, right, with the conventional method, and our new protocol you will find here the click chemistry for making this the 1, 2, 3 triazole that is recently, I mean, the couple of week ago, they accepted in the green chemistry as a wonderful outcome. We also prepared we wrote a review article, friends, here, how the Seven protocol is helpful to us to make cyclopropane. And you will see that first time that synthesis is accepted or graphical abstract in this manner. So you go with the reference and you find it and you get the huge idea. And another publication, European Journal of the Medical Chemistry, Chem plus Chem, ICS Bio and MedChem, right? And then the several things we have with the different journals. These are our published work. And we also received the RSS Sustainable Laboratory Grant Award from the Royal Society of Chemistry that was completed in the last couple of months ago. We completed, and this is our work, which is highlighted by the Malarial Wall, right? And you will find with the link. And we file a one patent, and I mean, it is approved by the government. and 4 more is there in the queue. I highly thankful to my Sardar Patel University because I love this university because we have taken huge support from my own university. I'm so proud of my university, and I will do better and better for my university in future also. So thank you very much to Sardar Patel University. Thank you very much to BKNMU and Naval Kapurai sir also for giving me this opportunity to little bit share our experience with the research scholar, right? So for that, I also thankful to the Royal Society because I associated with the RSC also, and then the ACS also, and of course I thankful to the SERB earlier. Now it is a ANRF. We received the one project from that, and we initiated the research work from this project support from by the government. So I'm highly thankful to my government and this IS and this ANRF now. So for giving us support to explore this kind of work. I also thankful to my collaborators. Friends, these are my research groups. Ruturaj Singh Wala from the Junagadh, Disa Wala from the Junagadh. This is Dipti from also. There are many of the people from the Junagadh. They are working with me, and they received a PhD degree also. Some of the students. This is my group. And friends, I discussed the sustainable approach. This is the father of the green chemistry. I met him. He invited me to meet and to discuss some of the things in the Delhi University with Professor John C. Warner. He's the father of green chemistry. So I had an opportunity in the 2023 to meet together in the Delhi University, and we organized a one RSC national seminar based on the funding we received from the Royal Society of Chemistry. So this is a little bit the moment with my scholar and my HOD, Professor Manish P. Patel sir, Kalisidan Mukherjee from the PD University, and the best, the organic chemist. I like this person because he's expert in organic synthesis. Kindly check his paper also, and you can inspire from them. So this is my family as well as the research scholar. So finally, thank you very much for your excellent support and inviting me to give this lecture. So friends, please let me know if you can have any other question. So thank you very much. Hello.
Yes, sir. There are no question from the audience,
Okay.
but it was a fruitful session, sir, from your side. Really, it's
So
a
it's my
great knowledge from your PPT as well as also presentations.
it's my pleasure, sir.
Yes.
Okay. So thank you very much, sir.
Okay. Thank you, sir.
So
Thank you very much, sir.
yeah, thank you, sir. Can I leave now, sir?
Yes, sir. Sure, sir.
Yeah. Thank you very much.
Pila leave